It's not my first slide, that's not right. Right. Okay, I'm Josephine Pemberton, since I've not been introduced, and here's the title of my talk. And I think that the quote from Nick, "For convenience, neglect non-genetic variants," is the point at which I'm taking off on this talk. Okay, so I'm going to talk about quantitative genetic studies on free-living wild populations. And this is a field in its own right within our topic of quantitative trait analysis. And I know it's a field because we now have a book marking that we are a coordinated set of researchers and problems. This came out a few years ago, 2014, and I'm using it as a starting point to emphasize the fact that this is a field which faces particular issues separate from a lot of standard quantitative genetic issues that come and arise from experimental studies and studies on farm animals. And it's also, I think, a very friendly and self-critical field which has gradually developed its techniques and approaches over time. And I'm going to show you a couple of slides from the first chapter in this book, which summarizes where we've got to, at least as far as 2011. So Eric Postma reviewed all the studies he could find that had estimated heritability of a trait in a wild population up to 2011. And what this slide shows is that there were a large number of studies going along at quite a slow rate in the early years, and then quite recently there's been a steep upshift, and that has continued to the present day. And I think we can attribute this rise to three different things, really. In these early studies of heritability, mostly they come from birds which have a pair of parents feeding chicks, so you can sort of infer who the parents are from the social pedigree. And a lot of these studies were using parent-offspring regression to calculate heritability. Three things happened in the sort of 80s, 90s era, which I think changed all that. First of all, in 1989, we learned about microsatellites, and they proved to be a major tool for establishing parentage in natural populations. Secondly, we had the uptake of the animal model for estimating heritability in natural populations. So this is the kind of modeling that allows you to do all relatives in a pedigree, not just parent-offspring regression. And it makes very efficient use of the data, allowing you to estimate quantitative genetic parameters with lower standard errors. And finally, we have methods for parentage inference that have statistical support, so using those microsatellites to improve the accuracy of parentage inference, of which Service, which we did in 1998, was one, but there are many others and better since. The other thing you can see from this slide down here is a rise in the estimation of whatever that black thing is, which is life history traits. So there's been a rise of interest in estimating the heritability of life history traits as opposed to morphological traits, etc. Here are some plots of the results. So what you can see, I'm only going to talk about these top two slides. Heritability estimates have actually, on average, declined over the period of study, as have the standard errors. So the heritability estimates, I think, very nicely prove Bill Hill's adage that the heritability of most traits is about 0.3. But both these trends that you see here in Eric's analysis seem to be to do with, first of all, the adoption of the animal model. So the animal model tends to generate lower heritabilities and also lower standard errors. And another component is that there are more and more life history traits being analysed, which means that, and they tend to have lower heritabilities than morphological traits. Okay, so as a field, this field has addressed many and interesting and diverse questions. And actually there are 10 questions listed in the introduction to this book that we're still all working on. But probably the one that interests me most and is the hardest is whether we can correctly predict and then accurately measure change in quantitative traits in the wild. And this question has an alter ego, which is, why do many quantitative traits show stasis in nature? So those are two opposite sides of the same question. And what we'd like to know is, can we make our sums work? I'm going to illustrate these attempts with examples from my own studies. So here's a map of Scotland. On this island here in the Inner Hebrides called Rum, we study a population of red deer. And in this island, St Kilda, out in the Atlantic, we study a funny little primitive domestic sheep gone wild, which is completely unmanaged. Both these studies adopt exactly the same annual censusing and study protocol. We basically catch as many newborns as possible. We follow them through their lives, so we have detailed information on their home ranges through their lives. We watch them in the mating season. Quite unusually for a wild animal, find them when they're dead. And we also have, and this is a difference between the populations, various other things we can study. So deer very conveniently regrow antlers every year and then drop them off. And we search for those antlers. We know who they come from because of the shapes, and also we can use DNA. And so we can study antler, quantitative genetics. On St Kilda, we're better able to catch the animals and phenotype them every summer. So this is how we weigh a SOA sheep. We just put them on a bathroom scale. In addition, both these populations, we've got quite good genetics going on. We've got 50k SNP chips for both populations and several thousand animals genotyped on those chips. And this allows us to establish very detailed multi-generational pedigrees. Okay, so returning to my question, the basis for how organisms should respond to selection is, of course, given to us by the breeder's equation here. So the response to selection is the heritability times the selection differential, and that is the core starting point to which I shall return at times in this talk. So the first estimates we made of selection on in my study populations was on St Kilda, and concern various morphometric traits, which are going to come back again and again, body weight and hind leg length over winter mortality events. And Jos Milner made these estimates about 14 years into our SOA sheep study. Oh dear, my microphone's fallen off. I'll just try and make that attach again. About 14 years into our study, Jos made these estimates, and what you can see from her estimates is that by and large, they are mostly neutral, as in no selection, or positive, with the positive ones coming at the right-hand end of these plots, which is when the density is high. So at high density, you get stronger selection. She also estimated some heritabilities for these traits, which you can see there, and they are non-zero heritabilities. And so based on the idea that you have positive selection differentials at least some of the time and non-zero heritabilities, you would perhaps be expecting to see a response of a positive nature over time, or perhaps you wouldn't expect to see any response on account of the slow rate of evolutionary change. In practice, what we have seen over our study is that the sheep are getting smaller. So this is a plot of lambs being weighed in August or having their leg length measured in August, and blue is males and red is females. Oh, this thing keeps falling off. Sorry about this. And what we see across many different traits on St Kilda is essentially all measures of body size are declining over time. So in other words, a simplistic analysis of how selection should be working in the population hasn't worked. And this is but one example, and perhaps a more extreme example, of a general problem in this field, which is that we're extremely poor at predicting the outcome of purported selection based on that breeder's equation. And the reason for this is perhaps, and I'm not going to delve into the details too much, demonstrated in this slide, which is the time series of the total number of sheep living on this limited island over time between 1985, when we started our study, and the present. So there are two things in here. One is that the population size bounces around like anything, and there are some years when there are nearly twice as many sheep as other years. And the second thing is we have an overall trend upwards in the population size on the island. It's actually 26 more sheep per year over 33 years. So to answer a question which has immediately leapt into your mind, the weight of sheep on St Kilda is going up. So the sheep are getting individually smaller, but the number of sheep is going up, and the overall weight of sheep on the island is going up. This serves to illustrate an absolutely, I think, crucial point about studying this problem in the wild, which is that the environment is continually changing, and in some cases it's changing systematically as well. So something's going on in this population to cause a change in the environment in terms of density, and there is ample scope for plastic responses in terms of the body size of the animals to overwhelm any predicted genetic response. And I should emphasize this can also be going on in space as well as time. So, returning to our breeder's equation, I'm now going to talk about some of the ways in which the field has had to cope with the things that the environment throws at our calculations. So thinking first about calculating heritability, over the years we have had to think quite hard about how to get the most precise heritability estimates out of our data. And some of the concerns that we have had have been listed here. So we might have missing and wrong links in our pedigrees, and we might have low depth of pedigree. And of particular concern has been the fact that we have many environmental effects which can masquerade as effects of relatedness. So I shall go into some of those in a little while, but there are aspects of the environment which can very much be correlated among relatives, making it particularly important to try and discriminate these variables. Speaking first of the pedigree quality, the main thing we've done within my projects to try and sort this out is to compare pedigrees. So we've got two pedigrees here. Number one is from microsatellite analysis, and number two is from overwhelming SNP analysis, and then comparing our estimates for the heritability with a genomic relationship matrix on very much the same animals. And if you look at the heritability estimates, which are in that column there, you'll see that we don't find much difference in our estimates between the three methods, a very slight reduction in the standard errors over many traits with the genomic relationship matrix. And the other approach that the field has also used very successfully is a simulation approach, thinking about what would happen if some of the pedigree links in a pedigree were wrong. And by and large, this supports the finding that most of our estimates are probably reasonably accurate with the pedigrees that we have at the moment, and small amounts of wrong pedigree links are not having a big impact. Moving on to the worry about shared environment effects, the first thing we had to think about in spades, really, was about the effects of different years having different, as it were, growing conditions. And this is an analysis of cast antler weight in red deer, showing that if you keep your eye on the heritability estimate, which is this lower light grey portion, depending on what else you put in the model, you get wildly different answers for the heritability. For example, if you put in permanent environment effects, if you put in the growing year of the antler, and if you put in the birth year, you get, in this best model, in terms of statistical explanatory power, you get an estimate of heritability which is 0.35 compared with other models with different values if you don't have some of those other variables in. So both the birth year of the male that grew the antlers and the year in which he grew the antler pair that you're currently studying have an effect on antler size and impinge on your estimate of heritability. Another major issue in our studies has been to think about maternal effects. So this now is a study of deer birth weight, and once again we've got different models being fitted with different random effects in according to a program of introducing more random effects in different models. And once again you can see that the heritability estimate, if you focus on the left-hand side here, the heritability estimate bounces all around the place, depending on what else you've got in the model. And putting in a maternal effect, so that is the identity of the mother, has a very substantial effect on your estimate. And on the right-hand side, what you're seeing here is that entering the maternal effect as two different random effects, basically the maternal genetic effect, so that's the mother's pedigree, and as a maternal environment effect, which is anything else explained by the mother's identity, greatly reduces the estimate of heritability. So presumably, because of the similarity in the pedigree relationships between a mother and her offspring, that is why this is going down a lot when you fit a proper maternal genetic effect. Finally, we worried quite a bit in our studies about whether the fact that animals live in a home range, and that home range has a specific habitat, and individuals overlap in their home range, we worried a lot about whether that might have an impact on our estimate of the heritability of a trait. So we have actually used our ranging behaviour of these animals to drill down into whether a random effect of home range sharing has any impact on our estimates of heritability. So in this set of models, which is for various traits in Soay sheep, the red is a random effect of space, so that's space sharing in most of these models. And we were interested in the extent to which it mattered whether you fitted the home ranging space sharing, whether that impacted on our estimate of VA, the heritability. To which the general answer seems to be not very much, but it is impacting, for example, if you look up here, on our estimate of the variance due to birth year. So there has been a lot of concern in this field about getting a really accurate estimate of heritability by controlling for all the other things that might be going on that might mimic relatedness. Of course, the Breeder's equation's got other bits to think about. Selection is a whole world of pain in itself to estimate selection accurately. One of the good things about the pedigrees that we've been able to resolve is they do give us an accurate estimate of the breeding success of every individual, so we're getting on much better than before we had those pedigrees. But we still have a number of topics of great interest. For example, I've already shown you some evidence that selection varies with the environment, and that has subcomponents of interest to study. And another issue that is very much to the fore at the moment is something called the missing fraction problem, that it's really impossible to study selection at the phenotypic level on individuals for whom you can't measure that trait. So various projects are afoot at the moment to try and get at that. But to think a little bit about the selection by environment interaction, this is just a modernized version of what I showed you earlier, which is that overwinter survival in young Soay sheep is, so the selection based on that survival is variable, dependent on density, and we have more intense selection going on at high density. And Darren Hunter and Michael Morrissey in St Andrews have recently pointed out that there are several possible subdivisions of components of this selection which are of interest. Namely, they pointed out that under different density you could have different trait means, different trait variances, different mean fitnesses, or different trait fitness relationships. And what they've actually shown is that the selection that we're seeing here is driven mainly by a change in mean fitness with density and some change, particularly in males, in the trait fitness relationship, which has implications for how constant we think selection is over time. There are also some issues which only arise when you put those two things together, the prediction side of the breeder's equation. So problems, or rather complications, that arise when you think about the heritability and the selection differentials together. Here are some of the issues that we are considering as potential sources of reinforcement or constraint in this section. So the first one, of course, is that both phenotype and selection are almost certainly multivariate. Secondly, that we could have selection by environment, which you've just looked at, in combination with genetic by environment interactions. And getting more complex and sophisticated yet, we could be interested in sexually antagonistic effects, either via genetic correlations or by selection, or this is subject of quite a lot of study at the moment, indirect genetic effects, either via genetic correlations or via selection, could actually reinforce or constrain what response we get in a trait. Just to consider the phenotypic, the multivariate question, it's very obvious that we should be thinking in a multivariate way about how things evolve, and we have a very nice multivariate breeder's equation telling us to consider selection on multiple traits, the heritabilities of those traits, but also the covariances, the genetic covariances between those traits. And we can certainly go out and study genetic correlations in natural populations. This is a set of genetic correlations done in the same study where we compared the genomic relationship matrix with pedigree estimates in Soay sheep, and we can certainly show high genetic correlations, luckily, between different ways of measuring leg length and lower genetic correlations between other kinds of traits. So we can certainly go out and measure genetic correlations. I didn't know the reference, but I do actually have a weightless elephant on a frictionless surface. I put this there because the key point in natural populations, which is different from what you do in laboratories or farms, is we don't actually know what is the target of selection, except for one thing. We do know that fitness is a target of selection. And so we can go out and measure traits like this, so foreleg, weight, hind leg, metacarpal, jaw length, all those things. But we don't actually really know which of those is the target of selection in reality. All we can do is measure what seems to be going on. And the main thing we do know is that fitness is the target of selection. So one innovation, which is basically based on writings of Price and Robertson in the past, is to deploy the thought of the genetic covariance between fitness and the trait in question as the way to predict the expected response to selection. So Michael Morrissey, who's in St Andrews, performed this analysis for various traits in Soay sheep and showed, for example, for body weight, this dark grey is what the breeder's equation would give you in its univariate form. So as I've shown you, a prediction of, or I've told you, a prediction of evolutionary change. A multivariate breeder's equation using the traits we can measure, also predicting evolutionary change. But if you use the covariance, the genetic covariance between fitness and the trait, the estimate for at least body weight is bang on zero, but of course with quite large standard errors. And the same, to some extent, goes for hind leg length, whereas you would predict a positive response in these two cases. Certainly, the Robertson-Price identity suggests that we either wouldn't see anything or we might, in fact, see a shrinkage. So we have much reduced expectation of the genetic response to selection from the evidence provided by the Robertson-Price identity. How am I doing? Okay. Okay, the other issue I wanted to deal with in this section, which is thinking about that h squared s side of the equation, and it's just one other example of the complexities there, is whether there might be a correlation between what's going on in the heritability under certain environmental conditions and what's going on with selection under certain environmental conditions. And this was first sort of demonstrated by Alistair Wilson when he was working here in Edinburgh with birth weight. So now on this plot, each of these dots is a year, and what he's done is to plot the selection differential on a cohort of lambs against the total heritability, and this now includes a maternal genetic effect as well as the heritability of the lambs in the lambs themselves. And it's not a very strong effect, but it is a negative correlation. And what that means is that in years when there is strong selection, the heritability is at its lowest. And in years when there is weak selection, the heritability is at its highest. So in principle, as an idea, this is going to constrain the response to selection. Now, this was quite an early study of its kind, and there have not been a great many since, but we recently thought to explore this general point a bit further in the other kinds of traits we study on St Kilda. And this was an analysis conducted, this was an analysis conducted by Adam Hayward, who has been in Stirling and is now at the Moore Dunn Institute. So a bunch of traits that we measure regularly in Soay sheep. Are they under selection? Yes. Does that selection get stronger at higher density? Yes, in some cases. Does it have significant additive genetic variance? Yes. Is there individual plasticity in the trait? So do individuals have different reaction norms in different densities? Answer, very often no, as it happens. And therefore, if you try to get out a G by E effect, it's either not applicable or no in every case. So although we appeared to have got something interesting going on with birth weight back here, I think that the general evidence for there being G by E effects, and in particular for them being coordinated in any way with selection effects, is not very strong. And there's been a recent meta-analysis which seems to confirm that across studies as well. In my final bit here, I'm going to talk about the response to selection, because the other side of all this is how can we measure the response to selection. I think I've shown you convincingly that just using the phenotype is no good. And there's a couple of approaches that have been attempted, which I'm just going to say a tiny bit about. One is predicted breeding values, and the other is genomic prediction. So, again, Alistair Wilson, some years ago, in demonstrating that there was a general decline in weight over time in all age classes in Soay sheep, also seemed to show that there was an increase in the predicted breeding values over time. So it's as if the animals were trying to get larger, but actually they were being stunted by other events. We now know that using predicted breeding values in this way is entirely inappropriate in natural populations. And I probably don't have too much time. I've gone orange here. But there are many criticisms of using predicted breeding values in natural populations, having to do with the problem that the predicted breeding value incorporates the phenotype of not only the individual under study, but its close relatives, and they tend to live in the same time period and hence the same environment. And so when corrected by Jared Hadfield, the actual expected evolutionary change here is well within the posterior expectation for drift. So no apparent expected response or measured response to the selection here. I just want to end very quickly on a new idea that John Slate is trying in Sheffield. We're going to hear, I'm sure, a lot more about genomic prediction later, but one idea is to use the SNPs and a training population to predict the genetic merit of a population and see whether the genetic merit of a population based on the SNPs is actually changing over time. And John would claim, and he's done various analyses to try and support this, that there is in fact a steady positive increase in the genetic merit for body size in Soay sheep over time. So to conclude, studying the evolution of quantitative traits in wild populations is a totally different ballpark to studying them on a farm or in the lab. The environment varies in time and space, affecting every part of the breeder's equation. Oops. The only trait we know to be under direct selection is fitness, and phenotypic change is not necessarily genetic change, and that's a particularly profound problem in the wild. And I'd just like to finish with my acknowledgements.
Josephine, for a fascinating talk. We've got plenty of time for questions. There's one at the front here.
Do you have any data on
Can you wait for the mic?
Do you have any data on the change in genotype frequencies over time?
Yes, we do. I mean, obviously, because we've genotyped animals over 30 years, we do indeed have that data. We haven't investigated it to the full yet.
Are there any indications that the story you're seeing in when you look at phenotypes is replicated when you look at genotypes?
John is doing that analysis at the moment.
Do you see large changes in error variance, the residual variance? Could you fit it year by year?
Could you say that again?
Do you see large changes in error or residual variance from year to year? Could you fit it year by year instead of just the general variance over the years?
Yes, no, I think we do, yes. Yep. I should just go back to Richard's question. We have found a couple of SNPs that have a large impact on leg length as a trait, but they are not changing significantly over time to the extent that we would hope. You know, they're not. We can't detect any significant change over time.
The number you quoted with an increase in population size of 26 sheep per year,
Yeah.
was that mainly through an increase in litter size?
No, it's coming about through increased survival of lambs in the winter. Yep.
It wasn't clear to me whether you've got complete ascertainment or whether there's any room for non-random ascertainment. Could your increase be due to change in behaviour? Do the small ones run faster, or the big ones run faster, or something like that?
It's a good question. I think there would have to be a correlation between intelligence and size for that to be true. We basically go out and do the same thing every year in terms of catching sheep.
Perhaps the smart ones have worked that.
Yeah. I think because we can see it also in the skeletal measures after death, regardless of whether or not we caught the sheep, I think it's unlikely.
This might be too obvious a point, but is there basically not very much evolution happening? So across the 30-odd years of the study, there just isn't, in both cases, there is very little evolution.
Yeah. But I guess that we started off by thinking, gosh, we've got some quite strong selection here, and we've got heritable traits, so we ought to see a response. And our observations are not unique. There are lots of other studies out there which would predict stuff that isn't happening. And so the chase is on for what are the mechanisms which can basically constrain what's happening.
So not only was Fisher right, but Darwin was right as well.
Thank you. This is a hypothesis for you, and it's really a case of your multi-trait problem. But essentially, the thing about being bigger that's an advantage is to be environmentally bigger. Being genetically bigger is actually no advantage at all. And if you had all of the relevant traits in your multi-trait, you would see this, but it's hard to do. How could you test my hypothesis that, if you've grown bigger because you got more food, that helps you. But if you're just genetically bigger, it's no help at all.
I'm not sure how one would get at that. There have been some attempts to look at the selection on the environmental component of a trait, but I'm not sure they've been entirely satisfactory in various ways, but I'll have to think about that one.
Are there any other questions? Okay, well, thank you very much, Josephine.
He's meant to be coming in. The technician is meant to be coming in.
Right, let's hope.
Good luck. The technician is meant to be coming in at the break. No, but between talks. I think escape is going to be the one. But no, the problem is it goes black on the screen, and then the mouse goes. Oh well.